Welcome to the problem-solving set on DNA as the genetic material. Problem 1. Carbon and nitrogen are greatly abundant in proteins, whereas sulfur is not. Why did the scientists Hershey and Chase use radioactive sulfur to label proteins in their experiments instead of radioactive nitrogen or carbon? So what was this experiment about? Remember that Hershey and Chase wanted to distinguish between protein and nucleic acid, or DNA, and they wanted to distinguish between the two to see what was actually the genetic material, because they knew that chromosomes were half protein and half DNA, and they didn't know which one actually held the genetic material. So let's go over what they used. Here they tell you that they used radioactive sulfur with a weight of 35 to label the proteins, and they used radioactive phosphorus with a weight of 32 to label the DNA. Now the problem is asking, why didn't they use radioactive nitrogen or carbon? Because nitrogen and carbon are actually more abundant in proteins, but sulfur isn't that abundant. Well, what makes up DNA? Remember that DNA is a carbon-based sugar, deoxyribose. So this has carbon. It has a phosphate group, which has phosphorus, and it has a nitrogenous base, which has nitrogen. So if they used radioactive nitrogen or carbon to label the protein, they would end up labeling the DNA as well, and there would still be no way to distinguish between the two. So they used sulfur because DNA has no sulfur. And they use phosphorus to label the DNA because proteins don't have phosphorus. So using this radioactive labeling, they distinguished between the two and saw that the DNA was actually going in the bacteria from the bacteriophage, or the virus that attacks bacteria. So that's how they determined that DNA actually held the genetic material instead of protein. Problem 2. Matt and Sasha are debating the details of Griffith's transformation experiment. Matt believes that the rough strain of Streptococcus pneumoniae underwent a mutation that converted it to the smooth strain that killed the mice, while Sasha says that the smooth strain passed genetic material to the rough strain. Which student is right? Well, let's quickly go over what was Griffith's experiment about. He had two strains of Streptococcus pneumoniae. He had the R strain, which was for rough, and the S strain, which was for smooth. So if you inject mice with the R strain, they remain healthy and live. If you inject mice with the S strain, they die. So what Griffith did next was he boiled the S strain, which in effect kills the bacteria, and then gave that to the mice. And these mice remained healthy and lived. And next what he did, he took the solution from the boiled S strain, added it to live R strain, and then gave it to the mice. And these mice died. So what happened? Here, the R strain, which in the beginning when it was by itself, didn't have any effect on the mice. They were healthy and they remained living. But when you added the boiled remains of the S strain, gave it to the R strain, and then injected that mixture into the mice, they died. So Matt believes that this R strain just went through a mutation that made it into the smooth strain that killed the mice. But Sasha says that the smooth strain passed on genetic material. Well, you should remember from the theory video from Professor Schmidt that actually here Sasha is correct. Okay? The R strain didn't just go under a mutation that made it lethal. It actually, what happened was, in the boiled solution, it contained DNA from the S strain. Okay? And when this was mixed with the live R bacteria, they went through a process known as transformation. And this is where a bacteria takes up naked DNA from the environment. Okay? So when the R strain of bacteria took up this DNA from the S strain, it actually ended up expressing the proteins from the genes and the proteins from this DNA, which made it lethal toward the mice. It didn't undergo some transformation, so for this case, Sasha is right. Question 3. While the chromosomal theory of inheritance was easily accepted, the nature of the genetic material was unknown for quite some time. Why did the determination of the chemical composition of chromosomes not provide enough information? Well, remember that this chemical composition of chromosomes that they're talking about is really that it's 50% DNA and 50% protein, roughly. So it's roughly half and half. Now that didn't give enough information because although we knew that the chromosomes were actually responsible for the theory of inheritance, we still didn't know if DNA was the genetic material or if protein was the genetic material. So our question now was, which is the genetic material? So this initial discovery of the chemical composition of chromosomes didn't really give enough information, and later scientists like Hershey and Chase experiment is really what found out that DNA is the actual genetic information material. But this initial chemical composition discovery wasn't enough. So that's the end of this problem solving set, and I'll see you next time.